Hi, everyone. I'm really honored to be here and been given the really exciting opportunity to talk to you what I deem sort of the flossing of environmental DNA, which is, how do we actually, it's so important to have standardizations and to do the science right, but how do we scale this up? It's not the most exciting, but hopefully I can be the hype man following the last two, talks about how NOAA Omics perspective, is going through and really pushing, eDNA into our operations. And first, I want to highlight that, I'm giving this talk, sort of on behalf of the NOAA Omics program, which has so many different members. I think there's over 100 scientists being represented, but also this is, hyper collaborative, and this is with folks at USGS. I heard of Steve Formel's online and Abby Benson, and really, folks at the Smithsonian. And this really comes from hours and hours and hours of conversations within the last year and a half, and really longer, moving forward, everything from the initial protocols, from how we're sampling, all the way through. And hopefully can give you a perspective on how we're trying to tackle this really big problem within NOAA. I also, amazingly, this hasn't come up yet today, but we are experiencing unprecedented ocean temperatures. And look at where we are. We're completely off the charts. My entire lifetime is represented on here, and we're in completely uncharted territories. But how do we generate this image that's both horrifying but also actionable and getting this made it to the front page of the New York Times, right? Like that's where the dream of science sort of goes, is it's showing up. You hear it on your podcasts, in the morning news. Well, this is a NOAA data set. And this is an integrated NOAA data set. It's a product that is being generated, has multiple staff members within NOAA that are dedicated to serving this. It has integrated observations from remote sensing all the way to Argo floats that are taking samples in the deep sea, many different kinds of thermometers, many different kinds of measuring temperature. We provide it global gridded daily. And they're referenced, and we integrate because we trust some of these sensors more than we trust other sensors. And so we calibrate them to each other. And I think this is where we're moving towards, because from a biodiversity standpoint, this is where our climate data is, right? And this has been pushed and really advocated for within our weather service and climate for decades, and this is where we need to move ourselves. Obviously we're starting maybe 40 years behind, but I think we can catch up because data science has moved a long way, right? And so we've spent NOAA as a collective, certainly not me as a new federal employee, and certainly someone who's never even taken an atmospheric science class, has spent an enormous amount of time and resources doing all of these, from standardized data, metadata formats, from specified workflows, from providing all of this data open access in real time or as real time as possible, really supporting FAIR and PAR principles and doing extensive calibrated integration efforts to make sure that these data products that are being served are trusted. And so that we can look at these data and we know that these are well calibrated and well validated. And so really my push that I'm suggesting here, and we take internally, very strongly within NOAA's, how do we push our biodiversity data so that we can make those same kind of products being served, so that we can, include this map, which is all the observations that have been made from GBIF and OBIS, but have that same level of quality assurance and quality control that we expect from our biodiversity data, and actually start getting eDNA biodiversity being served in these platforms, not just in the, last time I checked, there are a total of 40 eDNA data sets in OBIS and GBIF. Congratulations I know how much work it is, hearing from Catherine, what it takes to get it there. But collectively within this room, we must have over 1,000 different eDNA data sets, and all of those need to be mobilized into these platforms. So how do we do this? Well, we really need this biodiversity data mobilization. This was called out in the multiple strategies that we heard today. Obviously, this isn't just an eDNA problem. This is a problem for all of our biodiversity data, whether that's net tows or hook and line surveys or visual surveys. It was only until this October that the CalCoffee program was able to mobilize their faucet, their krill time series for 75 years, right? That's a 75-year krill time series that has not been made publicly available. Almost the vast majority of all the research funding that went to that was from the US government, so it should have been made publicly available. It takes a lot of time, research, and effort, and any of the data scientists that are here who are responsible for managing data know that this doesn't come for free, but we need this. We need this information because we saw that graph of how the ocean temperature is changing. You better believe that the biodiversity is responding to that, and we need to know how it is responding to that. And really, that's where eDNA has this incredible promise, because we can take a water sample, right? We can look at the species that are there. But really importantly, it allows us to co-locate our measurements in a way that other measurements haven't taken before, where literally from the same Niskin bottle, we can say, what species are we detecting? What's the carbonate chemistry? What's the pH? What's the temperature, right? And that allows us to answer these really critical questions of how biodiversity is responding to our changing ocean conditions, right? And so how do we do this? It's not going to be easy, and I've spent, operationalizing was actually in the title of my PhD. So I've been spending over 12 years now thinking about how we take this from an eDNA perspective moving forward, right? But it means end-to-end protocol and metadata reporting standards, because the folks that are doing the quantitative analysis, Ryan's in the room here, they want to know exactly what protocol you used, and you better tell them exactly what would they input on the PCR, because that's going to change amplification bias. And I'm sure some grad student 75 years from now who's also reanalyzing this data, because it's the fundamental baseline of what our marine ecosystems look like today, they really want that metadata standard. And so we also need to develop these mindful metabarcoding practices, do the right thing. We just heard about standards, guidelines, and best practices. I think in the metabarcoding space, really identifying what those performance standards and criteria are is really critical, and we need to move in that space. And I think we've learned a lot, but it's now time. That's one of the key things with this implementation. And obviously, we need to coordinate these mobilization workflows and integrate all these biomolecular observations, because that's really, to me, where we're going here is across NOAA alone, there's an estimate of multiple tens of thousands of eDNA samples that have already been taken. And they have never been collectively analyzed in one database, in one repository. But you can think of how many different research questions could be asked if all of that data was in one place. Scale that up to not just NOAA, across the entire US government, scale that up globally. That's what we need to be able to answer these questions at scale, because the fish that we're seeing where they are now is not where they're going to be in 5 years. And so one of the huge collaborative efforts that has happened, I see Luke and Catherine, who I know were really instrumental in this, but certainly conversations with Steve Formell and Matthew Biddle and many, many others in making this happen, is what does the NOAA data lifecycle look like? And how do we implement that? And so this came out in the NOAA data management guideline, which there's a QR code there for a link. Come talk to us if you're really interested. At least provide a baseline of what it would take to manage data within NOAA. What are those best practices internally for us? Hopefully this can be valuable for other folks. There's certainly a lot of black boxes that are listed up here that we have not quite solved yet, but I'll talk through a few examples of where we have spent a lot of time figuring this out. So one of them is the generation of study data templates. This is an effort really spearheaded by Catherine Silliman at AOML. But certainly many people contributing to this, with the idea of, okay, if we can put all of our data in the same data file formats, then we can write an R package or Python script that can take that and do what we need to do, sending the raw data and CBI, meeting the MixX standards, which are different than the Darwin Core standards, where we want our final data set to be. So the ability to crosswalk between these different standards that exist and allow for a simple integrated method. And we're looking for beta testers. So if people have data that they want to run in this and try to get their data sets on OBIS, please come talk to us. Our group, and a huge shout out to Han Weinrich and Shannon Brown on my team at PMEL, just went through the effort working with Katie Pitts and folks in Bari to generate these detailed protocols, start to finish, everything that you would need to do all of our lab protocols in-house, from field collection through PCR. We're still working on the rest of the protocols. It takes a lot of time and effort, but by generating these templates and examples, hopefully it's really easy for the rest of you to copy paste. And all you have to do is change the really tiny little things that you might be doing differently than what we're doing. At least you have an example to build off of. But this is really critical, because these are version-controlled accession. You can know exactly what we did in the future. These aren't going anywhere as long as you still have the internet. And a really big collaborative effort led by Katie Klimas and many other folks in this room was, what is the minimum information for publication of environmental DNA metabarcoding data, with this idea of we really need to keep track of all this information. So with this really came a checklist sort of building off of the ME checklist that existed for qPCR work, but how do we get that for metabarcoding data, and really allowing folks, when they're going to publish a paper, but also reviewers as well as journal editors, know, hey, this is what needs to be included, because there's a lot of papers that are out there. You read the paper and you're like, what primer did they use? And amazingly, it's not in there, right? And so that's wild, and so we can't let that happen. Stuff that is near and dear to my heart is thinking about how do we do the taxonomic assignment best practices, and we're going for bingo blackout, so I had to talk about completing reference databases. So we did this effort looking at monitoring programs within NOAA as well as NOAA collaborators in the Northeast Pacific. So this is all the way from the US-Mexican border, very extensive, what we're considering Northeast Pacific, all the way into the Arctic. And asking this question of what are the species you're actively monitoring you care about? And do they have reference barcode sequences? A third of them have no sequences to NCBI. None whatsoever. These are species that NOAA is actively, or NOAA collaborators are actively monitoring, right? That's kind of a problem. And obviously they're biased. They're biased to the things we care about. Sharks and whales, nearly 100%. Of course. Krill, we love krill. Krill have been barcoded. Mysid shrimp, absolutely not. Myctophids, oh man, it's brutal out there. And this is only for these two markers that we really care about. We were talking about the mitogenome scale, where we don't know what the best markers we might need, especially if we're going to be designing qPCR assays. The 12S is might not be the best place to go, and it's going to be different for every species. So if we want to bulletproof these things going into the future, we need to start thinking about whole metagenome sequences. We need to build these. But beyond just building them is how do we actually build these reference databases that we trust? We can enable trust in what they're doing, and I really think that the national strategy, pushes NOAA and pushes the federal government to be, this data broker to provide this information. And so we have, and folks here have thought about scheming up what it would take to have a curated reference database, so we can actually have taxonomic nomenclature curation, so we're crosswalking between all the different standards, because anyone who's tried to go from NCBI to worms and back knows it's a mess. For phylogenetic curation, we don't have, GenBake is a parking lot for sequences. How do we make that parking lot a curated parking lot? Building in some of these new tools. Lenore Pipes has this tool Tronco that allows you to make these trees for a single gene of interest at scale, which computationally would be intractable, but now is possible. And then also thinking about geographic curation, because a lot of work, including some of my own, show that use regional databases, are better at assigning taxonomy than a global database. And then building these tools so that if you can ask the question of, okay, well, these are the 15 species of fish that I care about in my backyard for my monitoring program. Which marker should I use? And my fish might not be is one of the better ones on the global scale. But if you really care about, surf perches in Southern California, it's a horrible marker. And Telio 12 works much better, right? So having the information is really, really important. And so another big thing that happens when we're thinking about these performance-based criteria is what's going to work for some applications is not going to work for others. So there's a really big resolution confidence trade-off. And so what we can see here is if you use this bootstrap confidence cutoff score, which is a specific parameter to the ANACAPA toolkit, but this applies to other classifiers, is if you get more lenient, you can increase the number of correct species assignments if you assign a reference database back to itself, but you also increase your error rate. And so the question is, you're going to get something wrong. Does it matter? And well, if you want defensible taxonomic assignments, especially if you're dealing with protected species or invasive species, right? And so you're going to use a completely different threshold for that than you might use for, say, a biodiversity survey where at a global scale, from a UN decade perspective, because we want what's best, but we'll accept a 1% error rate in assignment taxonomy because it'll come out in a wash. Whereas if there's a $100 million action on the line, whether or not you're detecting this endangered invasive species somewhere, that's a different question. You want that to be bulletproof, right? And so we need to be able to develop these defensible taxonomic assignments as a community. And I don't think we've done that yet, but that's going to be a key element of the implementation plan. And another element of that is within NOAA, we're actively working on doing a head-to-head comparison of all the different metabarcoding pipelines that we're using to figure out, can we develop a one NOAA, best practices, a single pipeline that we can use, or, at the very least, those performance criteria for this is working well. And another really big effort, that also Catherine had, counterpart AOML has been pushing really hard on is developing this EDNA to OBIS with many other collaborators, right? And if you have these metadata templates, can you go through and push it to OBIS? Anyone who's tried to do it knows that it takes hours and hours and hours, and it's a pain. But if we can actually build this software tool to make it easy, the only way everyone in this room is going to do it is if it takes under an hour. And so that's the dream, is that we can have this tool, it's easy to do, and make it so we can mobilize all this data. And thinking about this on a bigger picture, it's one thing to get the data into these global repositories. repositories. How do you take those global data from global repositories, not just the eDNA data, but all the visual data, whether that's from community science or citizen science, or whether that's from NOAA fisheries surveys, and how do we integrate that with our environmental data, whether that's from remote sensing or hydrography. And so this is an effort that we're doing, supported by NOAA, looking at data sets in the Arctic to integrate these things and plug them into existing NOAA tools that have already been developed, either by the NBUM program or the Integrated Ecosystem Assessment Program. And so this is just an example of the kind of workflows that we need to be developing to make these tools at the downstream end that everyone can use. And all of that is to support this big effort to think about integrating eDNA at scale. And so we have been through this really fun collaboration, West Coast Ocean Biomolecular Observing Network, which is endorsed by the UN Decade, bringing together as many different groups on the West Coast that are doing eDNA biomonitoring towards the goal of figuring out harmonization and standardization efforts, identifying those best practices with the hope that in the next few years we can actually start integrating. We ballpark there's about 20,000 eDNA samples on the West Coast. By the end of 2025, that could be synthesized into one giant time series. And they just did this in Eastern Australia, Brown et al. 2024, showing that by integrating them, they could see marine heat wave impacts. And this is the kind of data mobilizing that we want to get to, so that we can make and improve these maps that we have in our global repositories of biodiversity data. And so, this is really a shout out that with this strategy is to build this biodiversity data mobilization effort. We're going to need these standards. We're going to need, but a really big push for open access and traceable data, and really, ensuring that there's these extensive calibration and integration efforts so we can trust the data that's coming from different places, or when and where we can trust that data. So take questions.
Excellent. Thank you so much, Zach. Questions for Zach? Oh, we got a question. Can I get a mic? I see Kevin coming. Oh, but Susie's also running. Susie, we appreciate you. And you, Kevin.
I just don't want them to get hurt, Zach, really cool stuff. I actually, what really resonates with me, a lot of stuff resonates with me, what you said, but in particular, the need for mitogenomes as opposed to, barcodes. And I just, it's not really a question, it's more a comment. I really want to, echo how important that really is, because it sets the basis for our understanding of interspecies as well as intraspecies variation, but also it gives us the maximum design potential for new metabarcodes as well as new targeted assays, better, bigger, better, faster, stronger, all that kind of stuff. So any way that we can facilitate that is, in my book, golden.
Absolutely, and I've been a huge fan of all of your work, Karen. Because of that, we're excited to do that stuff. And the one thing we just looked into, just as an informal survey of asking sequencing companies, what's the price point differential? You get at least 20 times the data, depending on the size of the mitochondrial genome, for only 4 times the cost. And so we're looking at $150 per species to send them a vial of tissue. That's what it costs to get a full metagenome. And I'm sure you could maybe do it for cheaper if this was not a very scientific analysis. This was just us asking 3 labs, and I won't name them. And then also getting the price point, it's somewhere on the order of between 30 and $50 for them to do the extraction and get the barcode. So 100% we should be doing metagenomes. As much as I love frosted tips, 90s science needs to go, and we need to enter into the 2024.
On that note, thank you, Zach. Love it.